Hello everyone. Today, I would like to introduce a single-cell and spatial multi-omics integration and collaborative visualization tool called SGS. I will mainly discuss the background of software development, the main functional modules, and some visualization examples. First, let me introduce the background and significance of developing the SGS multi-omics visualization tool. In recent years, the rapid development of single-cell and spatial multi-omics technologies has enabled us to obtain multi-omics information at the cellular level, such as RNA expression, protein abundance, DNA methylation, chromatin accessibility, and spatial information. These technologies have revolutionized our understanding of gene regulation, epigenetic variation, protein expression dynamics, and cell interactions. Single-cell and spatial multi-omics technologies are widely applied in 1. Analyzing the cellular and spatial heterogeneity of diseases, 2. Constructing multi-omics regulatory maps, and 3. Conducting research in developmental biology, neurobiology, tumor biology, and other fields. In a recent study reported in Nature in 2022, researchers utilized multiple omics technologies such as single-cell gene expression, chromatin accessibility, and spatial transcriptomics to study different myocardial physiological regions at various time points in myocardial infarction patients and control groups. They constructed a comprehensive high-resolution map of human heart remodeling after myocardial infarction. Also, in 2022, researchers published the first spatiotemporal transcriptomic atlas of the axolotl brain in Science. This study performed spatial transcriptomic sequencing analysis on 6 developmental stages and 7 injury-induced regenerative stages of the axolotl brain, revealing the cellular and molecular characteristics of the axolotl telencephalon during development and injury-induced regeneration. This study provides important guidance for understanding mammalian brain regeneration. Driven by innovative platforms, diverse tools, and novel methods, single-cell and spatial multi-omics research have experienced unprecedented growth in recent years, generating vast amounts of multi-omics data. These data are characterized by diverse data types, large volumes, complex formats, and high dimensions. How to achieve joint visualization and collaborative mining of these massive, high-dimensional, and diverse multi-omics data is a significant challenge. Currently, mining single-cell and spatial multi-omics data is mainly divided into data analysis and visualization. Taking spatial transcriptomics as an example, the first step is data preprocessing, mainly image calibration, cell segmentation, and data quality control to obtain the spatial expression matrix. Downstream analysis mainly involves predicting spatially highly expressed genes at different spatial capture sites, inferring the cell type composition of each capture point based on gene expression, and further deducing communication and interaction networks between different cells based on existing cellular spatial annotation information. Data visualization is crucial for further mining more potential information. Currently, the main visualization contents of single-cell and spatial multi-omics include spatial gene expression distribution, spatial expression distribution of cells, tissue slice display, and molecular clustering display, such as a map-ETS-NE. For single-cell and spatial epigenomics multimodal data, it is often necessary to display chromatin accessibility, histone modification, and genome tract display. Data visualization is vital for single-cell and spatial multi-omics research. Researchers have developed various visualization tools which can be categorized based on their usage into command-line visualization tools, single-cell and spatial browsers, genome browsers, and comprehensive visualization tools. Command-line tools such as SIGNIC and ARCHR require high programming skills and are difficult to use. Existing single-cell and spatial browsers are mainly used for dimensionality reduction clustering, cell annotation information, and marker gene expression visualization, often limited to single-modal data visualization, unable to meet the needs for multimodal data visualization. Comprehensive visualization tools like Vitesse and Atlasplorer provide relatively comprehensive visualization functions, but require a complex configuration process for installation. Overall, existing visualization tools have limitations in terms of ease of installation, multimodal integration, and comparative visualization. It is imperative to develop new visualization tools that should be accessible, versatile, and capable of promoting in-depth interpretation of multimodal data within a unified framework. In summary, existing visualization tools have limitations in terms of ease of installation, multimodal integration, and comparative visualization. It is imperative to develop new visualization tools that should be accessible, versatile, and capable of promoting in-depth interpretation of multimodal data within a unified framework. To address the current challenges in visualizing single-cell and spatial multi-omics data, we have developed SGS. SGS Single-Cell and Spatial Multi-Omics Browser is a user-friendly, collaborative, and multifunctional single-cell and spatial multi-omics browser. SGS aims to provide research teams with a quick and easy-to-use cross-platform data visualization service hub. Through extensive data compatibility, it offers rich visualization modules, multi-dimensional comparative visualization, multi-panel collaboration, and exploratory functions to help researchers gain new insights from multi-omics data. Next, I will introduce the working concept of SGS. SGS is developed based on the Data Workstation concept and mainly consists of the SGS client and server. Users first need to download the SGS client and connect to a remote or local server through the client for SGS server installation. It supports cross-platform installation and use. Next, I will introduce the main functional modules of SGS. SGS not only provides a graphical installation interface supporting Linux, Windows, and Mac OS systems, but also allows batch upload management of data. Additionally, SGS offers multiple access methods and supports team collaborative exploration. SGS also has excellent data format compatibility, compatible with ONData, MUDATA, and various genome-mapped file formats such as GFF, VCF, BED, METHILC, GWAS, etc. Through the accompanying R package Saigon data, SGS can be compatible with data formats from mainstream analysis tools such as Sera, Archer, Signic, and Giotto. SGS supports the display of cell-to-D scatter plots, marker genes, metadata, cell proportion, tissue slices, and other annotation information. It also provides dual-screen comparative visualization functions for spatial multi-sample and multimodal data cross-modal comparison. The SC module SecMultiView supports multi-marker comparative visualization, offering various plotting styles such as violin plots, scatter plots, bubble plots, and heat maps. Furthermore, SGS has developed a new genome browser framework. It supports the visualization of genome data in various formats, including GFF, VCF, BED, BW, GWAS, METHILC, HIC, Long Range, BigInteract, among others. The dual chromosome visualization module in SGS supports the comparative visualization of omics data between two chromosomes, allowing interactive operations such as dragging, zooming, and scaling. SGS supports multi-panel integration and epigenomic multimodal data visualization, allowing users to conduct multi-panel synchronous exploration. Next, I will introduce the user case. SGS showed the CETROSEC data of mouse embryonic development spatial section in E9.5 period. Through 2D scatter plot and gene retrieval function, we can obtain the spatial plot of gene expression, gene co-expression module, and regulation of embryonic sagittal section in all periods, which is conducive to in-depth understanding of the dynamic differentiation process of cell groups during mouse embryonic development. In this case, we use SGS to display the 10x Visium multi-sample data of mouse brain. Through the SCUMPARE, SecMultiView, and the Tatchard functions, the spatial heterogeneity, cell proportion and key marker genes of the cells in the anterior and posterior brain slices were visualized and compared, providing valuable insights for exploring the type and status of the brain cells of mouse. SGS demonstrated the cellular heterogeneity of CG methylation signals of cortical RORB genes, as well as cell-specific CG methylation and chromosomal interaction signals of related genomic regions, and found that the chromatin interaction intensity was significantly enhanced and CG methylation signaling was weakened in the adult PFCL4-5FOX2O cell population compared with other cell populations, consistent with previous studies. SGS showed the heterogeneity of BLK gene expression cells and the corresponding gene structure and the associated cell-specific EQTL sites. It can be seen that its associated 5Es NPS related to autoimmune diseases are related to the expression of BLK and CD4NC, CD8ET, CD8NC, BMEM, and B in cells. Among them, RS-736336 mutation leads to the differential expression of BLK and B lymphocytes, indicating that it may play a role in the inter-individual variability of B lymphocyte tolerance. SGS displayed the spatial ATAC-seq data of the spatial chromatin accessibility landscape during mouse embryonic development, effectively integrated cell-specific chromatin accessibility signals through the SG module, and spatial distribution and expression heterogeneity of cells promoted the rapid acquisition of the epigenomic signals and spatial expression differences of target genes at the level of single cells. It provides a powerful tool for in-depth understanding of gene regulation mechanisms and expression patterns during mouse embryonic development. Finally, I would like to thank you once again for your support of SGS. We will continuously update the detailed user tutorials for SGS. Please stay tuned to our platform for the latest usage guides and case studies. For efficient communication, we have established the SGS Research Community Group. Feel free to scan the QR code to join. Come experience the powerful features of SGS and explore the limitless possibilities of multi-omics data.